we have started to talk about microscopy, and we talked a little bit about different, concepts underlying microscopy. Now I want to remind you, and I believe you did work with microscope on multiple occasions, just to remind you, what different parts of the microscope are. Look, yeah, you have to know what different parts of the microscope are doing. But one more thing that I wanted to do today is to remind you some practical considerations, let's put it this way. Not some fundamental, dry things, but what you should and what you shouldn't do when you do microscopy. So, eyepieces, come on, thing. Eyepieces, objective lenses, it's pretty obvious, right? Now when you do microscopy, remember to look at the magnification. In these microscopes, eyepieces or oculars, 10x. So to know the total magnification, all you need to do is to multiply magnification of each of the objective lenses by 10. Okay. The max magnification that this microscope can go to is 1,000. 1,000 will be this white labeled oil immersion lens. They oil immersion. They aren't supposed, you can use them without oil. The quality of picture will be terrible. When you use oil immersion, when you do microscopy, you go from scanning. I think, did we talk about, no? Okay. So you have 4 objectives. You have this red labeled scanning. You have yellow labeled low power magnification objective, red labeled high power magnification, and white label here oil immersion. You always start with scanning if you think that you see bacteria in the scanning objective. No, you don't. It's not bacteria. It's crap, it's bubble, whatever. It's not a bacteria. If you think you see it in the low power, it's probably not a bacteria either. They're still too small. You will see them, though, in the low power objective, using low power objective. You will see tiny dots. Yes, that's bacteria. But you're not going to see anything sizable. If it's more than a dot, like a period in a book, no. When you go to high power, 40x objective, the 400 magnification, the blue one, that's when you start to see actual cells. You can sort of figure out the shape and stuff. They're still going to be small now. If you want a really good picture, you have to go to oil immersion. The problem with oil immersion is that it works about 80% of the time. So make a really good sight with high power. Okay, I don't know, sacrifice an animal, pray, whatever your religion tells you to do, and then put some oil on the slide and slide this objective directly into the oil, so oil makes a bridge between the slide that sits on the stage and the objective. You will see a little bridge. This bridge prevents light scattering and increases resolution of the image. And then you have to be super careful in bringing your image back to focus, okay? Just because it may not work. One more consideration which I will remind you about, just telling you, when you put oil on the slide, avoid bubbles by any cost. Bubbles will mess up the picture, okay? What I do sometimes, I squeeze some oil, put it on a piece of paper so all the bubbles in that first drop, and then I move on. Stage here, it's called mechanical stage because you can move it with the knobs. Here, the knobs. It's very convenient because you don't have to use your hands and your movements are precise. In order to make it work, you may see that on the stage, I don't know how to show it to you better, there are these claws, the clamps here. Your slide should be stuck all the way into the clamps so it doesn't move, it's not loose. Then it's going to be super convenient to move the slide around. Focus knobs. Focus knobs, you see large ones. Those are called coarse focus knobs. These large coarse focus knobs are to be used only with a scanning objective because they move the stage a lot. Let's put it this way, okay? And if you use them at any other magnification, you're risking to break the lens. So at other magnifications, you use the fine focus. Okay? Why I tell you that you're risking to break the lenses? One more thing to consider when you do microscopy. Always look at the distance between the objective and the slide. I don't have a slide there, but you kind of get a gist. Say this is low power magnification. This would be about the right distance, okay? When you move to high power should almost, maybe, 1/10 or 2/10 of an inch be away from the slide. It's super close. When you slide oil immersion, it practically touches the slide. Because sometimes what happens, I have students having this, telling me they can't see anything. No shit, you can't see anything. It's too far. Just, I'm not trying to be dismissive, but before you think, oh, there is something wrong with my slide, it's probably fine. You did a fine specimen, but you're just too far. If that's the case, go back to scanning, start over. Does that make sense? The easiest way to find the object, to find the specimen, is to go back to scanning and start over. Make sense? One more detail that you actually will use. Do you see this tiny little lever? This one? This. I don't know how to show it better. This lever. That's the lever for the diaphragm. In this microscope, you can see it right here. It not only regulates the amount of light, with that amount of light, it regulates the resolution of the image. Generally speaking, when you move the lever, so you're looking from that side, all the way to the right, okay, the resolution is best, but image becomes pretty dark. So you have to fine-tune the image. You have to play with the diaphragm, you have to play with the brightness dial, which is right here. Just simply regulates the brightness of the lamp. Okay? When we're gonna do the microscopy lab, believe me, I'm gonna be running around helping you out and helping you to figure out how to do this. And I expect you to, be pretty good at it at some point. Okay? Deal? So let's talk about most common types of microscopy that you can encounter in microbiology. And for the purpose of the exam, you need to know 7 types of light microscopy and 2 types of electron microscopy. When I say you need to know, I mean I give you a description and you match a description with the name of the method. So we're going to talk about light microscopy first. Bright field microscopy is something that you will be using in this course. You observe specimen against the bright background. That's about it. Makes sense, right? So light shines on the background. You see the specimen, which is darker than the background. Dark field. There's some optical trickery that creates dark background, but cells are actually lit somehow. I don't really know. It's physics, so it's not my piece of cake, so I don't know how to do it. But there are microscopes that allow you to observe the image that is lightened up against the dark background. This is really useful to look at the live specimens, especially live specimens of really thin bacteria. Does that make sense? Here you can see spirochetes of Borrelia burgdorferi. They're really, really narrow, really thin. But this dark field allows you to better visualize them. So far, we're good. Now, phase contrast. So this is where magic happens. When I say magic, I don't really mean like an actual magic, but a famous British sci-fi writer, Arthur Clarke, once said that very advanced human technology is indistinguishable from magic. So for me, it's indistinguishable from magic. A bunch of optical devices in the microscope use such phenomena as diffraction and interference to produce this really high contrast, high resolution images, which can be used to observe live unstained specimens. This microscope does not have a capacity to produce phase contrast. This one does, and I will try to mount some sample when time comes and show you what phase contrast is. I'd say it's overhyped. I mean, it's cool, but not like incredibly awesome. Okay. Differential interference contrast, or DIC, is actually a development of phase contrast. It uses even more sophisticated optical equipment to produce what looks like three-dimensional appearance of live specimens. This method allows you to identify certain structures inside the cell. The bright field microscopy won't let you do it to a large extent. We're good? Fluorescent microscopy. Now, as you go from bright field to phase contrast to fluorescent microscopy, the price of equipment rises, obviously. These microscopes run, I think, about if you took AP1 or AP2 in these new classes, these new microscopes there, they run for about $2,000. This one will be probably about $4,000 because it has phase contrast capacity. Fluorescent microscopes run at you can buy used for about $10,000. New will be $20,000, $30,000. Okay, so what do? They have a UV light that lighten up the specimen, and in response to that UV light, you know the black light? That's the same idea. In response to that UV light, specimen can start to fluoresce by itself, or sometimes scientists may use special dyes, special reagents to turn the specimen into being fluorescent. Have you heard about glowing rabbits or glowing fish when scientists modified? I think you can buy it now at the pet store. Fish that produces green fluorescent protein, and it will fluoresce in the black light in the aquarium. That's basically the same idea. You have a protein that, if exposed to black light, starts to fluoresce. Scientists actually made fluorescent mice and fluorescent rabbits. Now, it's all fun and whatnot, but in applications, in scientific applications, it is extremely useful because you can specifically label cells of interest with highly specific dye and see them fluoresce. One example that is used in applications such as biological and quality control in pharmaceutical companies. I mentioned mycoplasma. Remember we mentioned this microorganism as the one that doesn't have cell wall. Mycoplasma is exquisitely small and it's really hard to see it using conventional bright field microscopy. This one doesn't work with mycoplasma. So what you can do, and mycoplasma is a scourge of various biotechnological processes. It can contaminate all kinds of biological systems. So in order to detect mycoplasma, you can take a bunch of human or animal cells that you cultivate. You can add a special dye that interacts specifically with mycoplasma. Does that make sense? Then you wash, do all these technical steps, and then you put your microscopic specimen with object that you control under the fluorescent microscope. And if there are any mycoplasma cells, they will start to lighten up green. And you can say if your culture is contaminated. Okay. Now, fluorescent microscopes, often labs buy them, research labs. So they are reasonably priced. Let's put it this way. We were using old fluorescent microscopes when we were doing experiments on mice. We had 3 of them, so feasible. Now, confocal microscope is kind of a Mercedes-Benz of microscopes, light microscopes. This is one of the most terrible images of confocal microscopy that I've seen in my life. But I guess this is taken from that free online textbook that I told you. Perhaps that's the one that they got their hands for free. So what does confocal microscopy do? It uses a laser to do a cellular tomography. You know what CT scan or MRI are, right? Those machines take multiple quote-unquote slices through the human body, recreating a 3D image, 3D structure of the human body. Does that make sense? Confocal microscope does the same. It takes multiple slices in multiple planes at different angles and then combines those slices together to recreate a 3D image of the cell. This picture does not represent it at all. Confocal microscopes are fairly expensive. I believe the lowest price for those is about 50 grand. So usually this equipment is bought for the department or more often for the institution. There can be several microscopes placed in so-called core facilities. So when you need it, you go there, you pay, you use it. That's what we had in Louisiana. You pay, you use it, you get images, you go back. Because majority of people don't need them every day. Makes sense, right? And finally, if that was Mercedes, this is, I don't know, Rolls-Royce, Lamborghini of microscopes. This is called 2-photon microscopy. Now this is, this is magic at its best. I have no idea how this works. I will never be able to understand the physics of this. I can tell you what it does though. You can take a live specimen, any live specimen, from a bacterial biofilm to a thin slice of animal muscle. If you can, anesthetize mouse and slide the muscle under the objective. And this method will allow you to visualize, individual cells moving. So it allows you to shoot a movie about cells, floating around, moving and stuff like that. And you can label those cells with fluorescent dyes. And you will see some cells will be green, some cells will be blue, some cells will be red. So you can have multicolored image And you can distinguish saying, okay, this is, I don't know, this is Pseudomonas, this is E. coli, and this is Bacillus, whatever. Does that make sense to you? So this is really top-notch, state-of-the-art technology. The only problem is that it costs a lot of money. When I was working in cardiovascular physiology, the department where I was working at, they bought one of these 2-photon microscopes. And because they used it on mice all the time, so there was a line to that microscope. And I asked the price. The microscope itself was about 50 grand. It's normal for a good fluorescent microscope. The most expensive part was the laser, okay, that lights up those cells. The laser cost $250,000. I was shocked. It's like, holy cow. We have that amount of money? But it produced absolutely unbelievable images and great movies about the cells. Of course, it's not just to produce movies. It's much easier to see how the cells interact, where do they go, you know, all that kind of stuff. Now, those are 7 types of light microscopy. Any questions? I believe the question that you have, what would be the exemplary question in the test? Something like, this type of microscopy produces a 3D image of the cell, confocal. Does that make sense? I will make it really, really descriptive, really thorough, and I never had any problems with those questions. They're really clear. If you know, you know. I want you to know 2 types of electron microscopy. And as I'm talking to you, I realize I probably need to update this at some point. So this is transmission electron microscopy. In transmission electron, so first of all, what is the difference between the light microscopy and transmission microscopy? When we shine the light, what do we call the particles of light? Do you know? Photons. Photons, yes. So these photons run through the sample, they get refracted, they get reflected, their trajectories change, right? In electron microscopy, basically the difference is instead of photons, we have a beam of electrons. So these electrons run through the specimen and they get refracted, they get reflected, and then electrons that pass through get detected by whatever device. Usually it's based on the photoelectron effect. They get detected and software analyzes the pattern of electron refraction, let's say. Does that make sense? So basically it's sort of a special eye, electronic eye, that allows to analyze how trajectories of electrons were changed. Does that make sense? Okay, think about this. You have something in front of you. You have no idea what it is. And you start shooting water at it. Okay? And water gets all splashed out. You obviously cannot explain what is it there that, you know, produces that kind of splashes, but you get the gist. So those electrons splash in all different directions. So in transmission electron microscopy, electrons go through the specimen. Now, what does it allow you to see? It allows you to visualize the internal structure of the specimen. This is one of the famous transmission electron microscopy pictures. It was taken, I believe, in 1972 by Frank Murphy. It's an image of Ebola virus. Now, you can see that this image is two-dimensional, truly two-dimensional. It's a section. Do we agree on that? You can kind of see what's inside the virus, but you can't really fully appreciate its form. You understand what I'm trying to say? You can't really tell if it's perfect circle like a sausage, or maybe it's a little bit flattened. You understand? I usually compare transmission electron microscopy to like when you cut a stick of salami and you look at the inside. You don't really know how long the stick is, but you know what's inside. Did that make sense? So transmission allows you to see what's inside. Now, scanning electron microscopy allows you to see what's on the surface, what is outside. If you look at these cells of Campylobacter jejuni, you have no idea what is inside of those cells. But you have a pretty good grasp on their shape. Does that make sense? That clear? So basically, if we keep comparing microscopy with a stick of salami, scanning electron microscopy is just looking at the stick. You know the size, you know the shape, you have no idea what's inside. Makes sense, right? Now, if you would Google scanning electron microscopy images, you will see perhaps the most beautiful images taken by science ever. If you Google transmission electron microscopy, those are most unremarkable. They're really boring, but they're really valuable. Okay? Why I said that this should be updated. Nobel Prize, either for the last year or year before, was given to cryo-electron microscopy. I'm not going to ask you this, but to give you an idea how far we moved from 1899, when Ivanovsky identified a virus as a filterable agent, never seeing it or something. So what basically happens in cryo-electron microscopy, you can take a sample and cool it down, really cool it down. Like, I'm not sure that it's liquid helium, but for sure liquid nitrogen. And then you use electron microscopy technique to look at the object. But the resolution, so this is an individual cell, right? Right there. Using cryo-electron microscopy, you can look at the individual virus and see individual subunits of the virus capsid, the structure that surrounds it. You can almost see individual, well, I think now with some high resolution, you can see individual amino acid residues in the proteins. It is unbelievably cool, unbelievably powerful, and what's important, you can use a pretty dirty sample. You don't have to have a high purity sample. So, to give you an idea how science progressed, in, say, late 80s, early 90s, if people figured out the structure of a protein or structure of the virus, that would be a thesis project, and that would be 1 or 2 publications in the most prestigious journals, like Cell, for instance. If now people figure out the structure of the virus, it's a boring Tuesday. Okay? So, like, okay, we got another structure. Sure. It's a paper, yes, but nothing like too incredible. Okay? That make sense? Now, let's move to the concept of staining. Sorry. Staining of the specimen. So, first of all, how do we do? We're going back to the basics, basic microscopy. We're going to do wet mounts a lot. It is fundamental technique and it's super simple. You take a microscopic slide, take a drop of water, mix your bacteria in. That's your wet mount. What you can do with it? You can put the a cover slip on it, and that's going to be your live specimen. That make sense? Stick it under the microscope, look at it, you see bacteria moving around. You can't really get much out of it. They're moving around, so what? Maybe you can observe them moving towards the increased concentrations of chemoattractant, but only if you add one. Now, bacteria are moving. That's one problem with observation. It's much harder to look at something that moves. And the second problem, remember we talked about contrast? How bacteria are pouches of water, and we have to observe them against watery background. In order to increase contrast, we stain the specimens. In order to stop specimens from moving, we fix the specimens. Fixation can be chemical, or it can be done by temperature. So on the left here, illustration 1, you see somebody not lazy fixing a specimen on the heat pad. We're going to use much simpler and faster method. We're going to be using Bunsen burners. You prepare wet mount without a cover slip and run this wet mount through the Bunsen burner 3 times. 1, 2, 3. Specimen is fixed. Okay. What is also important, not that you just killed microbes. That is cool. Yeah, they're not moving. But you also fried them so they stuck to the slide. Now you can add dyes. You can wash them. Well, not fire hose them, but reasonably wash them. And they will stay at the slide. That makes it super convenient. Now, we're going to do a lot of staining. And stains can be of 2 major types. It can be simple and differential. Simple stains will stain all microorganisms alike, no matter what their structures are. Are we clear? If you stain Bacillus anthracis, E. coli, or Mycobacterium tuberculosis, they will all appear the same. Differential stains allow you obviously to distinguish microbes based on their different properties. Maybe internal structures like endospore, can be external structures like flagellum, or it can be a structure of the cell wall. Let's talk about simple stains for a second. So positive stains chemically, they consist of a molecule of a dye that charged positively. And they stain negatively charged surface of the cell. Now, let me ask you this question. Can you think of a cell membrane? Can you? Cell membrane? A bacterial membrane? Okay. What is the charge of the cell membrane surface in bacteria? Let's figure it out. Cell membrane. What does it consist of? Which molecule?
Lipids. Phospholipids.
Phospholipids. What sticks outside? Which residue? Which part of the molecule sticks outside? Hydrophilic. Which is what? Which part, more specifically?
Polar.
Chemically, it's
Phosphorus.
phosphate. Yes, it's phosphate. It's phosphate anion. And anion means that this phosphate is charged how? If it's anion. Negative. So all those phosphate heads are negatively charged. Does that make sense? Anything that is positive will stick, be attracted to those negatively charged phosphate heads. Does that make sense? So positively charged chromophore positively charged dye will highly efficiently attach to the negative surface of bacterial cell, producing this positive stain. Is that clear? Now, negative stains have chemical that is negatively charged. We clear? Now, negatively charged molecule and negatively charged surface of a bacterial cell, how would they interact? Would they attract? Would they repel each other? Repel. Two negative charges would repel. Does that make sense? So, it turns out that the stain is in the background. Bacteria is not stained, but you see how contrast this image is. This is called a negative stain. So basically, positive stain stains the specimen, while negative stain stains the background. Now, differential stains, more interesting. They can stain cells according to the presence of different structures, like flagellum or endospore, or the structure of the cell wall, like a Gram stain. And we're going to talk about the stains right now. So this is simple stains. Only two types that you need to know for the exam are positive and negative. I will not ask you to distinguish between basic and acidic. Do not ask, okay? Don't worry about it. I want you to understand, though, if a stain colors the cell, it's positive. If a stain colors the background, it is negative. Am I clear? Am I clear in my expectations from you? Say it one more time.
There's no difference between positive and negative.
Yes, that is correct. Positive and negative. I'm not asking you about basic versus acidic. You have to understand that if I use positive stain on 15 different microorganisms, they're all going to look the same because it's a simple stain. Say they're all going to look blue if I use crystal violet. Does that make sense to you? Now, come on in. Differential stains. These allow you to tell different microorganisms apart based on structures, that they have, structure of the cell wall, and so on and so forth. First that should come to mind is Gram stain. We will talk about procedure in 3 minutes, but remember when we talked about cell wall in bacteria, Gram stain allows you to distinguish these two big groups of microorganisms. Gram positives with a membrane and thick peptidoglycan cell wall, and Gram negatives, the membrane, thin cell wall, and the outer membrane. Why I put so much emphasis on the Gram stain? I mentioned before, and I want to reinforce it again. It is a really simple technique. But this is actually one of the to-go methods in the clinical microbiology. I listened to an interview with the head of clinical microbiology lab at Stanford University School of Medicine, the medical center. It's associated with Stanford University. And she was telling about all different sophisticated types of equipment that they use to analyze large volumes of microbiological samples. And then she was asked, okay, it's 2 a.m. in the morning and there is a blood sample, not blood culture, blood sample from a patient who is really, really sick. Blood culture is already started, to cultivate it, but you need to do something. what you're going to do, let's say we're going to do gram stain. It's going to give us the information whether the patient has gram-positive or gram-negative infestation. And based on this, physician can direct specific type of antibiotics. At least that gets the treatment started. Does that make sense? Then you can do all the fancy stuff. We got it? I also mentioned when we talked about cell wall that some microorganisms, such as Mycobacteria tuberculosis, cannot be stained using gram stain approach because they have a layer of mycolic acid on the outside. So acid-fast stain is used to stain Mycobacteria, used to stain Nocardia, and other acid-fast microorganisms. In this image, you can see clumps of red acid-fast Mycobacteria compared to non-acid-fast blue cells. Does that make sense? So this would be used to stain mainly for causative agent of tuberculosis. It can be used if somebody is suspected to have leprosy, because Mycobacteria leprae is also acid-fast. Endospore stain, well, it allows you to identify microorganisms that produce endospores. Can you remind me, when do endospores form? In what type of environment? Harsh. So first of all, it means that these microorganisms that form endospores can survive a pretty severe environmental insult. Does that make sense? Not only this, there are two most common clinically common genera of microorganisms that make endospores: Bacilli and Clostridia. So if you stain your sample and you see endospores there, you can say it's either Bacillus or Clostridium. That's it. If they gram-positive, that's it. If they are gram-positive and they make endospores, that's either Bacillus or Clostridium. And if you're in the hospital and you have Clostridium, and it's a lot of it, and the patient has diarrhea, that's C. diff. Does that make sense? So this, you don't really have to do sophisticated analysis anymore. Okay, you're done. Like, it can be done pretty fast. With Clostridia is a little bit more complicated because they are anaerobes, but kind of leave it behind. Flagellar stain. In this Bacillus cereus, you can see tiny little flagella. Now, think about the size of flagellum compared to the size of the cell. I'm not talking about the artist representations, but realistically speaking, you look at bacteria, they're super tiny. Flagellum, just flagellum without any stains or anything. Would you be able to see them using light microscope? No, they're too thin. So how do you visualize them? What's the principle in flagellar stain? If something is too thin, what should you make it to see it? Something is thin, you got to make it thicker, yes. And we do it by adding the chemical that sticks to flagellum and just makes it thicker. Does that make sense? And finally, capsule stain. That is super clinically important because it allows us to identify capsules that form this extra protective layer around bacterial cell, protect them from phagocytosis. And if we're dealing with some microorganism and we stain it using capsular stain and we see that it has capsule, we can expect it to be more virulent. Am I clear? Do you understand why it's clinically important? Say you have a patient who's infected with Streptococcus pneumonia. fine. It's treatable. You take a sample, you run capsular stain, it's non-encapsulated. You can breathe. It's going to cause disease, but probably not going to kill the patient. If it's capsular, that's bad news. They have really huge difference in virulence. So encapsulated Streptococcus pneumoniae is extremely virulent and very deadly. Does that make sense? Now, I can ask you about the difference between simple and differential stains. I can ask you about difference between positive and negative stains. I can ask you about gram stain and acid-fast stain. Guess what? There are 3 stains that I'm not going to ask you about because it doesn't make any sense, which are endospore, flagellar, and capsular. Because if I ask you, what does capsular stain stains? You tell me capsule, duh, right? It's the answer is in the question, so obvious. Make sense? Any questions about stains so far? Now, here's the procedure of the gram stain. The procedure that we're going to do, the procedure that you will learn. I want to show you how this differential stains work. Acid-fast stain conceptually is the same, but we're not going to talk about it. We're going to talk about gram stain. So imagine that you have a mixture of gram-positive and gram-negative microorganisms. And at the first, you do a wet mount of your sample. First of all, you fix it. You will see why. You fix it. So all the cells are dead, they're not moving, and all the cells are fried to the slide. We clear? Then you add crystal violet. It's a stain, a dye, that stains all the cell blue, or purple, whatever you want to call it. Gram positives, gram negatives, they're all blue. Does that make sense? You wash away crystal, you let it see it, you wash away crystal violet. Remember, you can wash your cells now because they are fried and they stuck to the slide. Right? You're not going to remove them. And then you add the chemical known as iodine. It's not like really an iodine, but that's solution of some iodine compound. This is called mordant. So what mordant does, it basically kind of precipitates, modifies crystal violet so it cannot be easily removed. I usually explain it slightly different. It's not correct explanation, but it allows you to better understand the logic. Basically, iodine makes crystal violet stick to the cell wall. And crystal violet, after this step, sticks to the gram-positive cell wall really well. But gram-negative, not so much. Does that make sense so far? Your cells are still indistinguishable. They're all blue. Any questions so far?
So it sticks really well to the gram positive.
Yeah, but gram-negatives, not so much. And then you wash it away, blah, blah, blah. You add alcohol. This is the most important and the most tricky step. What alcohol does, it is decolorizer. So it washes away crystal violet. But remember, iodine made crystal violet stuck in gram-positive cells. So if you do this step correctly, crystal violet will be washed out from gram-negatives only. Does that make sense? This is why it is a tricky part. Because if you a little bit overdo alcohol, it'll wash away crystal violet from everything. So that's kind of an iffy part. Makes sense so far? Good? Now you have two types of cells on your hands. One is colored. It's still blue, right? And one is not because you removed all the crystal violet. So what you're supposed to do? You're supposed to add what is called counter stain to stain gram negatives. And in this procedure, counter stain is safranin, which is red. So what you end up with is the mixture of blue or purple gram positives and pink or red gram negatives. Does it make sense? The whole idea. So you stain everything the same, then you treat it so one type of bacteria holds on to the stain better than another. Then you wash the stain off, you wash it off only from gram negatives, and you counter stain gram negatives with red color. And you've got these two wonderful microorganisms on your hands. In real life, for instance, for gram positives, it's a little bit tricky. You're always going to remove some crystal violet from them. So it's going to be a bit of a spectrum of blue / purple / really dark purple violet. I don't know, all the spectra from dark purple to bright violet. With gram negatives, it's easy. Always pink. Okay? Any questions? I want you to know the steps and the function of each chemical. So what is the function for alcohol? It's decolorizing. Clear? Good? There we go. Another pathogen. Bacillus. Really fascinating one. Some of it you hopefully will never encounter. Some of it, unfortunately, you will encounter many times in your life. So we're going to talk about 3 species. Bacilli are endospore formers, first of all. They are very resistant to the environment. Not only this, they actually exist in the environment. They're everywhere. Remember when we did a hand wash lab and there were large blobs? That's Bacillus. Somebody picked up the spore. Next week, as I told you before, you're going to do sampling of the environment. If you find some dry space that was not touched by humans for a long time, like top of the cabinet, one student once sampled the sink in the middle of a table, which wasn't used, I suppose, for like a year. It was laden with bacilli endospores. They're everywhere. Okay? So that makes them really resistant to the environment. And the first species that I want to chat with you about is Bacillus cereus, which produces thermostable toxins. Let's decipher. What is toxin? If I ask you a question and I don't say that it's complicated, then it's simple. What is toxin? Poison. Great. Brilliant. It's poison. Bacteria produce poisons. Okay? Bacterial toxins, just another poison. What is thermostable?
Doesn't denature with heat.
Yeah, if you keep it warm, it's not going to denature. It's going to still be a toxin. Now, Bacillus cereus is found in the environment. And remember, it produces endospores. Now I'm going to give you a description of a process. You have endospores of Bacillus cereus that are exposed to the water, organic chemicals, and, well, some warmth, about 50 to 60 degrees Celsius. What's going to happen to endospores when they are exposed to water and organics? Remember that word starts with G, like plant, huh? Germinate. Endospores will start germinate, and they will produce an actual microorganism. Does that make sense? And this microorganism will start to reproduce, and no surprise, it's going to produce a toxin. Now imagine all of this happens in the rice cooker. You have a bunch of rice, you pour it in the cooker, you cook rice, okay, and then it's an Asian restaurant, so you're not going to make a new batch for every client that comes in, right? You're going to have a giant vat of fairly hot rice to serve it to your clients. The temperature is about 50 to 60. For Bacillus as the vegetative cell, it's not super comfortable. It will reproduce and eventually die. But toxin stays there. And germination happens when you mix this fairly dusty rice. You know, it's like rice is not clean. Make sense? When you mix this rice with water, endospores germinate. You germinate, microorganisms produce toxin, die, but toxin is still there. People eat rice and get food poisoning. It was described multiple times. It is associated with specifically keeping the rice at this 50 to 60 degrees temperature. Does that make sense? I'm not saying that you shouldn't eat in Asian restaurants. We didn't talk about helminths that you can find in sushi yet. Anyway, since it's food poisoning, antibiotics don't help. You understand why? Yeah, you don't swallow any bacteria, it's just a toxin. So all you can do is, fluids and lay down. Usually, Bacillus cereus food poisoning is fairly mild, plus for 1 to 2 days. That much. Now, Bacillus anthracis is one of the few microorganisms that was seriously considered to be a biological warfare. It is a causative agent of anthrax. There are 3 main forms of anthrax: cutaneous, here and here. You can see that this is a fairly mild and limited form. This wound or lesion is called eschar. In this case, this patient has a massive necrosis of the forearm. Now, cutaneous anthrax is treatable, not self-resolving, but it's not going to kill you overnight, basically. Enteric and pneumonic forms are severe, enteric being the most severe. Sorry, not enteric, pneumonic being the most severe, respiratory anthrax. When you inhale endospores or bacteria, that's really bad. So what is the main transmission cycle? Now this image overrepresents it. If I would be able to draw a cow, I would make an image myself. But if I would draw a cow, you would have nightmares. So I have to steal those images online. How people get infected. They get in contact with infected cattle or other large hooved animals. In countries that are endemic, countries where anthrax is common, like China or India or Central Africa, some parts of South America, in these countries, anthrax is transmitted from cattle to humans. In countries where anthrax is sporadic, meaning that it happens every so often, Transmission happens to a large extent from caribou to humans. It happens in the northern regions. Okay. Now, transmission happens by contact with infected cattle, and yes, cows can die of anthrax. Okay. Sometimes, very infrequently, transmission can happen after the consumption of a meat from infected animals, and it can be transmitted from large, herds of cattle via respiratory route. That's the worst part. So far, you with me? Now, why it is so hard to get rid of anthrax? So imagine that you are in the northern part of Russia, which is not very different from, say, northern Canada, like Northern Territories. Indigenous people that live there in northern part of Russia, traditionally they herd caribous, and that's their source of meat, milk, skins. They trade that stuff, very, like, I would say, primitive farming. They have enormous herds, thousands of caribous. Now, if animals in this herd get sick, you cannot just kill them and sell the meat, because meat is dangerous, right? So what you're supposed to do with all those thousands of caribous, ideally, you have to slaughter them. What are you supposed to do with corpses? Huh? That's what is done.
Burn.
Ideally burn, but if to burn a human, you have to produce a lot of heat. To burn a caribou, you have to produce even more, and the oven, or whatever they call it, should be even larger, because the animal is larger. Now, it's like in the middle of nowhere. I mean, it's really interesting idea for a startup to provide services of the mobile crematories, but I think the clientele is not going to appreciate it like very often. So what they do, instead of burning, you cannot just put them in a pile and set them on fire. It's not going to burn them. You have to burn them to ashes because of the infective nature of the microorganisms. So they bury them. They dig a giant hole in the ground, they put them all in, they cover it, and they put signs. Biological hazard. Don't cross, don't let animals to graze, and blah, blah, blah. It's in tundra, so people don't really live there. They just move around. Eventually, those signs will go missing, and at some point, animals will start grazing. In the meantime, bacilli that were in those caribou will form endospores. Caribou will rot, okay? Those endospores end up in the soil. That's tundra, permafrost. When it gets warm, permafrost melts. Well, partially melts because it's permafrost, but it partially melts, and water from the soil starts to go up using capillary forces, taking endospores with it. And at some point, endospores end up on the grass, animals eat it, new outbreak of Bacillus anthracis. Those outbreaks, I know for sure, in Russia, in that northern region, they happen every couple of years. The last one was a year and a half ago. About 300 people got infected. One kid died. It was a pretty sad story because his grandmother didn't realize it was anthrax, and he ate some meat, caribou meat, got enteric anthrax and died. Nobody else died. So they called a lot of animals, buried them, so expect another outbreak in some time. Okay. Now, so far, any questions? Yes? Yes. Yes, it does. It does. So, the question was, does the enteric form, one that is consumed in the gut, replicate inside the humans? The answer is yes, it does. So they replicate in subcutaneous tissue, they replicate in the lungs, they replicate in the gut. So you treat them with antibiotics, yes. Now, what about biological warfare? The only time when anthrax was used in some kind of a operation that looked similar to biological warfare was anthrax attacks of 2001. You may remember that scare when several people received envelopes with lyophilized Bacillus anthracis. It was a white powder. It got aerosolized, they inhaled it, developed respiratory anthrax, and several people died. Now, a couple of things about that story. First of all, it turned out that was a scientist. Disgruntled, allegedly that was a disgruntled scientist from CDC who was refused a promotion or something, some kind of, employment issue. And he was working with anthrax, so he packaged it and sent it out, and then he killed himself. When investigation got to him, he realized that it's inevitable, and maybe it wasn't him, but it kind of, lines together very nicely. That was the only time it was kind of actually used. Second outcome, now all postage in the United States, all packages, all mail, is actually checked for anthrax. Which you can say is impossible, but actually is possible. Company that makes machines that run analysis, I believe they make millions of dollars. Okay, they make machines, they make all reagents. So those automatic machines, they collect samples from all mail in the United States and do quick analysis for anthrax spores. Okay. Anthrax was considered to be a biological warfare. It's probably the most feasible, because spores are very stable. You can spread them, you can pulverize them, you can package them in a bomb, but it's another problem with biological warfare. First problem is the delivery method. How you spread it. Do you want to use it locally? Do you want to use it to a large scale? Second, probably issue that is often kind of ignored, willfully ignored. Let's say country A wants to use biological warfare against country B. The question is, what's the purpose? To kill everybody? Well, okay, maybe you will achieve that aim, okay? So killed everybody. If you use something like anthrax, something like Bacillus anthracis, you make an entire country forever endemic for anthrax. The entire country is contaminated. It's going to get into animals. It's going to get into people. And why do you need these spoiled territories? That's a good question. So biological warfare is, I don't believe in it being a warfare. I totally believe in it being a terroristic warfare, but that's a different story. Now, other species worth mentioning, the Bacillus subtilis, the most studied gram-positive microbe, very commonly used in research. Now, Bacillus thuringiensis. Anyone does organic farming? Organic. Do you do BT?
Not that I know of.
Insecticide?
It's
actually-
not necessarily me. It's like my boyfriend. He's like all
Okay.
organic with everything. And we had stuff that he ordered online that
To-
the post office did the poke test on.
To grow?
Mm-hmm.
So BT is certified organic insecticide. You can use it on your plants, according to requirements from whatever oversight organ regulates this. BT is the toxin produced by Bacillus thuringiensis, and it kills a lot of diphtheria, like mosquitoes and flies and a lot of them. I believe what happens, it changes the pH in the insect gut. I don't remember from acid to base or from base to acid, but it changes the pH completely, and they can digest food and they basically die. Okay, BT is used by in Florida, actually, they have a special service, the state service, that spreads BT over the southern tip of the state and the 7 Keys. After they had an outbreak of several mosquito-transmitted diseases, they really cautious about it, and they control the mosquito population there. They have a fleet of planes and helicopters, and they fly over 7 Keys and Miami and spray BT. It's safe for humans, okay? Absolutely no side effects, so they do it.
And animals as well.
Yeah, it affects insects. Again, as far as we know, but all experiments showed that humans and animals have no biological response to BT. Okay, make sense? So we're going to take a break, just logical. When we'll come back, we will review the quiz, look at the results, and chat about culturing the microbes.